Tim Johanson is today's speaker. He tinkered with Drosophila as an honours student in the Department of Anatomy across the road. And when he came to interview, he sort of said that he had H1 of 91, so didn't know much immunology. I thought, oh, 91. Anyway, at the time, we, Len Harrison and I were agitating to get Mark Chong back from New York. So it sort of coincided that Mark Chong came back with all his microRNA, not only know-how, but his tools, the Drosha and Dyson knockouts. And so it was sort of opportune for, that we thought, well, microRNA seem to be involved in regulation of so many things. We thought, well, maybe they would be involved in the differentiation of DCs. As it turned out, it wasn't DC differentiation so much, but DC development, as you'll hear about from Tim. Tim's sort of pretty green-fingered. I think he's touched on a lot of techniques that required some pretty fine touches and did them with aplomb. I think one of the other things about Tim is that his approach has always been quite measured. So that's in not just science, just throughout his life as well. So he's more likely to try to convince you than try to hustle you. He's processive rather than headlong. And he's quite thoughtful, both in a colloquial sense as well as in an academic sense. So the story with the DC sort of took different turns, and I think it was a great credit to Tim that he really managed those turns and actually succeeded. So, Tim.
Very well done. Thanks, Andrew, very much for the very typical introduction. Given the rather unusual choice of photograph used of myself to advertise today's seminar, I know many of you are in attendance today to not necessarily see me, but in fact the charming giant that is Jason Siegel present today's seminar. So to those amongst you in this category, I apologise. And for the perhaps deception, and the only solace that I hope to offer you is that I hope that your celebrity disappointment will not be followed by scientific disappointment. So as such, I'd like to present to you today the most coherent story to come out of my PhD, and that is in identifying a microRNA-independent role for the nuclease Drosha in the regulation of myelopoiesis, or the formation of myeloid cells. So, why is it, given that we all in the room share the same 20 or 30,000 genes that make up the human genome, that some of us end up looking like slightly malformed Hollywood celebrities, while others amongst us look relatively normal? The answer to this question is the multiple layers of gene regulation over and above simply the presence of those 20 to 30,000 human genes. One of those layers is made up by microRNAs. And since their discovery in nematodes in the early 1990s, they've gone on to become arguably the hottest small RNAs family of the past 2 decades. As such, they're a family of small endogenous RNAs that play their gene regulatory role through the targeting of messenger RNAs, or mRNA. And they play this regulatory role through the targeting of mRNA, as I say, for translational repression and ultimately degradation. As such, they've been shown to target a vast array of genes, as I suggested, and as such target and play a role in almost every cellular process examined. As such, we can see that represented in some way here in this microRNA targeting network analysis, which shows all of the potential interactions of these series of microRNAs with a set of mRNAs. So as you can see, a vast regulatory potential. So the biogenesis of microRNAs is a stepwise process that relies on 2 key enzymes. That is Drosha and Dyson. It begins with the transcription of a microRNA gene, which goes on to lead to production of a primary microRNA. This primary microRNA contains either one or multiple double-stranded RNA stem loop structures, that the stem of which is recognised and cleaved by Drosha, leading to the production of what is known as a pre-microRNA. In the cytoplasm, this pre-microRNA is then recognised by the second enzyme crucial to this process, known as DICER. This leads to the production of a mature microRNA duplex, one strand of which goes on to guide the gene regulation that I aforementioned. So Drosha and DICER fall into the family of the RNase 3 family of enzymes. This family of enzymes is an ancient family that's found in bacterium, yeast, flies, and then obviously in higher organisms. The family is defined by the presence of an RNase 3 domain, which imparts the unique ability to cleave double-stranded RNA. This is just a representation of a bacterial RNase 3 enzymes bound to a double-stranded RNA. And the important point here is that this binding brings the RNase 3 domains into contact with both strands of the RNA, and thus leading to the cleavage of the double-stranded RNA. So when I began my PhD, as Andrew suggested, nigh on 4 years ago now, we wanted to look at the role of Drosha and DICER, and as such microRNAs as a family in this process, that is hematopoiesis, or the formation of the blood and immune systems. As you can see represented here, hematopoiesis begins in the bone marrow with a hematopoietic stem cell, or HSC. As hematopoiesis progresses, daughter cells increase their commitment to a particular lineage, and thus moving through a series of progenitor stages, eventually go on to give rise to all of the one of a vast array of terminally differentiated immune cells, such as these or these. So as is subtly suggested here by colour, this hematopoietic system can be broadly broken down into two arms. That is the myeloid and lymphoid arms. And then, as Andrew suggested, when I began my PhD, the role of microRNAs was reasonably well understood in a number of these lineages, for example, in T and B cell development. However, our knowledge of the role of microRNAs, and in fact Drosha and DICER, in this pathway, namely the development of dendritic cells, was almost entirely unknown. So a fairly obvious target for a PhD. So if we focus more specifically on dendritic cell development for a moment, as the previous schematic suggested, it begins in the bone marrow with a hematopoietic stem cell, or HSC, which can go on to give rise to either a common myeloid progenitor, or CMP, or a common lymphoid progenitor, CLP. Common myeloid progenitors then in turn can go on to give rise to a common dendritic cell progenitor, or CDP, which in turn can give rise to pre-CDCs, or pre-conventional dendritic cells, which in turn obviously gives rise to the CDC, or conventional dendritic cell. CDPs are also able to give rise to plasmacytoid dendritic cells, or PDCs, and this is an ability they share with a common lymphoid progenitor. The two mature these dendritic cell subtypes then go on to maintain and regulate an immune system that is both responsive to immunological challenges, but also appropriately self-aware, so as to avoid autoimmune issues. And thus, dendritic cells regulate and maintain a healthy and properly functioning immune system. So just to give you a bit of an idea of the journey I'd like to take you on today, I'll give you the entire idea of the known roles of RNase 3 enzymes in hematopoiesis as we currently know it, and all of this revolves around DICER. I'll then take you through some of the work we've done identifying an impact of Drosha or DICER deletion on myeloid cells and dendritic cells more specifically. And then, as Andrew suggested, in fact the title suggests, I'll then take you through a rather unexpected microRNA-independent role of Drosha in dendritic cell development, and we believe myelopoiesis more broadly. So the first indication of the importance of RNase 3 enzymes in hematopoiesis came in an early study identifying that DICER-deleted HSCs are outcompeted by their wild-type neighbours. As such, if DICER is deleted specifically in HSCs, it's shown that in vivo they are outcompeted by their wild-type counterparts, and that is suggested by their reducing percentage of the total hematopoietic system that is derived from these DICER-deleted HSCs as opposed to from their wild-type neighbours. More specifically, RNase 3 enzymes, and again primarily DICER, has been looked at in myeloid cell development. And it was shown that if DICER is deleted specifically in these early myeloid progenitors, that this alters the differentiation potential of these early myeloid progenitors. This was further characterized by a block or reduction in myeloid cells observed in vivo. The only published paper to look at the role of RNase 3 enzymes, and again I suggest just DICER, in dendritic cell development was this one. And what they did was to delete DICER specifically in cells that express the integrin and typical dendritic cell marker CD11C. As you can see here, CD11C begins to be expressed at a relatively late stage in dendritic cell development, that is, after the common dendritic cell progenitor stage. And what Cubers and colleagues showed was that deletion of DICER specifically in CD11C expressing cells had no impact on either the numbers or, in fact, function of conventional dendritic cells or plasmacytoid dendritic cells in these mice. As was alluded to, in fact, by Cubers and colleagues, they suggested that, in fact, the CD11C deletion system is inadequate for examining the role of microRNAs and, in fact, DICER in dendritic cell development, simply because the half-life of a typical microRNA is, in fact, longer than that of a conventional dendritic cell or a plasmacytoid dendritic cell. As such, these dendritic cells will die long before full microRNA deletion is ever achieved, and so thus the impact can never be observed. Given that inadequacy of the CD11C deletion system and the fact that all of these papers only looked at the role of, did not verify that the deletion of DICER was in fact due to the loss of microRNAs, which could simply be done if you think about that the biogenesis pathway requires both Drosha and DICER. If you delete Drosha in a similar manner to that which you delete DICER, you should be able to confirm that the impacts of DICER deletion are in fact caused by the loss of microRNAs. Given that this was not done, the question still remained, are microRNAs important in dendritic cell development? And the way that we went about answering this was to look at the impacts of deletion of Drosha or DICER on myeloid cells, and in fact hematopoiesis more broadly. In order to avoid the autogenetic restriction of promoter-driven deletion, we used a tamoxifen-inducible deletion system. This system relies upon the ubiquitous expression of a fused CRE-R molecule that is inducible by tamoxifen. Upon the treatment of tamoxifen, this fused CRE-R molecule goes on to delete floxed or LOXP sites flanked genes, in this instance, Drosha or DICER. Importantly, all of our controls contain this ubiquitously expressed CRE-R molecule as well. So one of the early experiments we did to try and identify the role of both Drosha and DICER in hematopoiesis was a mixed bone marrow chimera experiment. For the non-immunologists amongst you, this involves taking bone marrow from control, Drosha floxed or DICER floxed mice and mixing them with distinguishable wild-type bone marrow. This bone marrow mixture is then taken and transferred into a lethally irradiated mouse. This lethal irradiation destroys the hematopoietic system of this mouse, and thus only the bone marrow and in fact the HSCs that are derived from this transferred bone marrow give rise to the hematopoietic system of this mouse after a 10-week reconstitution. After this reconstitution period, we then did a 3-week tamoxifen treatment and then analysed various different cellular subsets within various different organs at that time. So what this system allows us to do is to delete Drosha and DICER specifically in the hematopoietic system of these mice, because the only CRE-R expressing or tamoxifen responsive cells in this mouse are derived from this transferred bone marrow at the origin of the experiment. So if we look in the spleen of these mice, which is a readily available and large reservoir of dendritic cells in the mouse, and we simply collagenase digest these spleens and stain with antibodies against Siglec-H and CD11C, we can identify two populations of dendritic cells. That is the double positive plasmacytoid dendritic cells, or PDCs, and the single CD11C positive conventional dendritic cells, or CDCs. And as you can see in the control mice, about 0.1% of this spleen is made up by plasmacytoid dendritic cells, and about tenfold this amount is made up of conventional dendritic cells. So the first really interesting finding was that when we delete DICER and examine the DICER-deleted dendritic cells in the spleens, we can see that there's a dramatic reduction in the percentages of both PDCs and CDCs in DICER-deleted spleens. However, and perhaps more interestingly again, when we examine Drosha-deleted dendritic cells, we can see that there is a more dramatic reduction in the Drosha-deleted CDCs and PDCs and CDCs compared to DICER or in fact control. So a more severe impact was observed in splenic dendritic cells when we deleted Drosha to that when we deleted DICER. And this is quantified here in that you can see a reduction when we delete DICER, but a more dramatic reduction when we delete Drosha in both CDCs and PDCs. Now, this phenomenon wasn't restricted to dendritic cells, in fact, and it was more broadly observable in myeloid-type cells, that is, granulocytes and monocytes. However, interestingly, when we examine cells from the lymphoid arm of the immune system, that is T and B cells, we can see that at least over this 3-week deletion period, T and B cells are relatively unaffected by the deletion of Drosha or, in fact, DICER, suggesting potentially that myeloid cells are more dramatically affected than lymphoid progenitors. Now, in order for us to say anything meaningful about these experiments, obviously we have to show that the deletion of Drosha and DICER are equivalent in these cells. As such, we use qPCR to show that the deletion of Drosha and DICER in these cells is approximately equivalent. However, furthermore, what we did was to go on to look at the functional effect of this deletion on, for example, on a panel of microRNAs. So as you can see here, when we delete DICER or Drosha, we see an equivalent effect, functional effect, on a panel of microRNAs due to that deletion. This allowed us to say that both genetically and functionally, the deletion of Drosha and DICER is equivalent in these cells. This allowed us to say for the first time definitively that microRNAs do have a role in myeloid development and in fact dendritic cell development. However, the more severe phenotype that we observe when we delete Drosha to that when we delete DICER suggests perhaps something more interesting, in that it suggests a microRNA-independent role for Drosha. That is because, and the logic behind it is, that if Drosha functioned exclusively in this microRNA biogenesis pathway, then we would expect to see similar, if not identical, phenotypes when we knocked out Drosha to that when we knocked out DICER. Because we see a more severe impact of Drosha deletion than DICER deletion, this suggests a role for Drosha in addition to and outside of this role in microRNA biogenesis. And that's a point I'll return to in a moment, and fairly obviously. But the next question that we wanted to ask was at what stage of dendritic cell development are Drosha and DICER playing these crucial roles? So in order to do this is just a reminder of dendritic cell development, but in order to do this, we return to our mixed bone marrow chimera system. However, in this instance, instead of examining the terminally differentiated dendritic cells in the spleen, we in this instance examined the proportions of these progenitors in the bone marrow of these mixed bone marrow chimeras. So if we take the bone marrow of these mice and we remove most of the mature immune cell types, which we term lineage-depleted bone marrow, and we stain this with antibodies against c-KIT and SCAR1, we can identify this double-positive LSK, that is lineage marker negative c-KIT and SCAR1 double-positive population, which is enriched for hematopoietic stem cells. So in our control, we can see approximately 4.5 or 5% of this lineage-depleted bone marrow is this hematopoietic stem cell enriched population. A similar proportion is seen when we delete DICER. However, when we look in the Drosha-deleted bone marrow, we can see that there's an approximate doubling of the percentage of the bone marrow that is this LSK or HSC-enriched population. If we quantify this, we can see that, again, there's this approximate doubling of the Drosha-deleted LSKs compared to either DICER or control. And this suggested to us a block or a defect in the development of potentially myeloid cells or hematopoiesis more broadly. If we go on and move further down the dendritic cell development track and look at the percentage of the bone marrow that is CMP, that is common myeloid progenitor, again we see this approximate doubling of the percentage of the bone marrow that is CMP. However, in this instance, if we compare to DICER, we also see this increase in the DICER-deleted bone marrow. Now, this is consistent with previously identified roles of DICER in myelopoiesis. And in addition, because we see this overlapping phenotype when we delete Drosha and DICER, suggests that this impact is in fact due to the loss or the discontinuation of the production of microRNAs, because we see the same phenotype when we knock out DICER to that when we knock out Drosha. If we move down the other path, that is towards the lymphoid, we see again suggesting potentially that the lymphoid arm, at least over this 3-week deletion period, is unaffected by the deletion of Drosha and Dicer, that we see this non-significant change in the percentage of the bone marrow that is the common lymphoid progenitor. If we move to the final stage of dendritic cell progenitors in the bone marrow, that is the common dendritic cell progenitor, we can see that we begin to see the phenotype that we observe in terminally differentiated dendritic cells of the spleen. That is a more severe reduction when we knock out Drosha to that when we knock out Dicer. However, in this instance, we're observing that impact in the bone marrow as in common dendritic cell progenitors. So in order to further dissect at exactly what stage Drosha and Dicer, or in fact Drosha, is playing this crucial role in dendritic cell development, we turn to an in vitro development system, and that is using FLT3 ligand, which is a growth factor which is well understood, a lot of the work done here at WEHI, to drive dendritic cell development in vitro. This experiment was based upon the sorting of LSKs, CMPs, CLPs, or CDPs from either our control or our Drosha-deleted bone marrow. As I say, we then cultured these in FLT3 ligand, and then at the end of that time point, examined the dendritic cell number and percentages derived from these 4 different progenitors. And the primary finding here was that Drosha is only playing a critical role in dendritic cell development at a very early stage. As such, what we saw is that if we examine the percentage of these cultures that is CDCs and PDCs in the LSK-derived cultures, we can see a significant reduction in the percentages of both CDCs and PDCs in this culture, suggesting an important role for Drosha in this developmental process. In contrast, if we look at the percentage of CDCs and PDCs that is derived from the common myeloid progenitor that are have Drosha deleted or not, we see no significant change in the percentages of CDCs and PDCs, suggesting that once a dendritic cell reaches a common myeloid progenitor state, it can quite happily continue to a mature dendritic cell in the absence of Drosha. The same thing is true if we examine the percentage of PDCs. In this instance, we could never detect CDCs in CLP-derived cultures. We can see that, again, PDCs can quite happily develop in the absence of Drosha from a common lymphoid progenitor state. And perhaps unsurprisingly, given the results from the CMP and CLP cultures, when we look at those cultures derived from the common dendritic cell progenitor, we can see that we see no impact of deletion of Drosha on these cells. Importantly here, what I won't show you today, but importantly here, we did show that the deletion of Drosha and in fact microRNAs are equivalent across these 4 culture systems. So, just a brief first summary. What I've shown you, and I hope I've convinced you of, is that Drosha deletion has a more severe impact on dendritic cells than does Dicer deletion. Furthermore, this impact functions at a very early stage of dendritic cell development, that is prior to the CMP or CLP stage. What I haven't shown you today, and perhaps strengthening the suggestion that this is a block and defect in development, we've also shown through a number of different experiments that this impact of Drosha deletion is not due to significant changes in cell death or proliferation of these cells. And then finally, and importantly, what we suggest is that this impact of Drosha is likely due to a microRNA-independent role for the RNase 3 enzyme. So what are the microRNA-independent roles of Drosha? All of them involve and revolve around the binding and or cleavage of double-stranded RNA stem loop structures similar to those that are recognised and cleaved in primary microRNAs by Drosha during the production of microRNAs. The first and in fact best defined of these microRNA-independent functions of Drosha was the recognition and cleavage of stem loop structures in messenger RNA, leading to degradation of this mRNA and thus playing a gene regulatory role, a direct gene regulatory role. Another and perhaps more contentious and at this point debatable microRNA-independent role of Drosha is in the processing of pre-ribosomal RNAs and thus Drosha being crucial in the production of ribosomal RNAs. But as I said, slightly contentious this one. Drosha has also been shown to be able to target and cleave double-stranded RNA stem loop structures in retrotransposon-derived RNAs, and thus playing a role in genome surveillance and thus minimising the potential harmful impacts of these harmful mobile genetic elements. In the only cleavage-independent microRNA-independent role of Drosha, it's also been shown that Drosha can bind to stem loop structures upstream of particular genes, leading to, in an unknown manner, enhancement of the transcription of those particular genes. And then the final known microRNA-independent role of Drosha is in the production of DNA damage-induced small RNAs that play some function, and thus Drosha plays some function in the assistance of DNA repair. So the question is, which of these known or potentially known microRNA independent roles of Drosha is responsible for our block or defect in myeloid development that we observe upon Drosha deletion? Given that we see a differential impact or a non-overlapping impact when we delete Drosha to when we delete DICER, that suggests that the underlying phenomenon or the causative mechanism behind this is a role not shared by Drosha and DICER. By that logic, we rule out microRNAs as being a causative mechanism of this effect that we observe in myeloid development. Furthermore, we can also rule out these two microRNA-independent roles of Drosha because these two are also shared functions of the other RNase 3 enzyme, DICER. As I suggested in the beginning, the best defined of the remaining known microRNA-independent roles of Drosha is the cleavage of mRNA leading to the degradation of this mRNA. As such, the first mRNA shown to be directly cleaved by Drosha was called DGCR8. And this phenomenon was first observed as a consistent increase of the levels of DGCR8 mRNA upon Drosha knockdown, but not upon DICER knockdown. And through a very elegant and highly controlled study, Hahn and colleagues showed that the underlying mechanism of this was, as you may have guessed, the cleavage of double-stranded RNA stem loop structures within the DGCR8 mRNA by Drosha, and these are shown here. Within months of this study, a number of other studies showed that this mechanism was conserved in many different cell types and in fact across many different species. So in order to determine whether this or one of the other known or in fact unknown microRNA-independent roles of Drosha were causing or underlying our myeloid defect, we turned to a microarray. And what we did was to sort LSKs from control, Drosha-floxed or DICER-floxed mice. And the reason that we sorted LSKs is because we've shown that this microRNA-independent role of Drosha is functioning at this very early stage, and thus if we're going to catch a glimpse of it, we need to look at this very early hematopoietic progenitor. We kept these LSKs for 72 hours in vitro to allow full and in fact equivalent deletion of Drosha and DICER, extracted RNA and ran a simple microarray. And the focus of our focus was on transcripts that were differentially expressed between Drosha and DICER knockout LSKs. The logic being that these guys would be, the result of their upregulation or downregulation would be due to a microRNA-independent role of Drosha. Those that were equivalently changed compared to control in DICER and Drosha-deleted LSKs could be said to be caused by the loss of microRNAs. And this is just the results showing the normalized levels of all of these transcripts in DICER-deleted LSKs deleted LSKs compared to DICER-deleted LSKs. And if we ran a simple differential expression analysis, we could pull out those guys amongst these that are differentially expressed between Drosha-deleted and DICER-deleted LSKs. Of the approximate 40, in fact, the exact 41 transcripts that were differentially expressed between Drosha and DICER-deleted LSKs, approximately 75% of these guys were primary microRNAs accumulating in the absence of Drosha. This is to be expected, as that if you can see, if we remove Drosha, these primary microRNAs will not be able to be processed and will thus accumulate. They will not be processed by DICER because they are not in this pre-microRNA form and thus will accumulate. So if we ignore these green primary microRNA transcripts and just focus on those that are non-microRNA related, we can begin to potentially pick out the guys that are causing or underlying our microRNA-independent role of Drosha in myeloid development. Of these guys, the only down-regulated transcript was fairly expectedly Drosha, but however, there were no other down-regulated transcripts when we compare the levels in Drosha-deleted LSKs to DICER-deleted LSKs. This fairly logically suggests that this microRNA-independent role for Drosha is not playing a part, not having an impact in LSKs in our system. In that, if this was having an impact, we would expect to see a number of transcripts down-regulated upon the deletion of Drosha because they would no longer be enhanced by the presence of Drosha. Furthermore, if we examine the ribosomal RNA precursors that have previously been identified to be a target of Drosha and thus important in ribosomal RNA biogenesis, we see there's no impact or no change in the levels of these precursors in dicer-deleted LSKs or, in fact, in Drosha-deleted LSKs, suggesting again that this is not a function, microRNA-independent function of Drosha that is impacting in terms of LSKs. That leaves one, and in fact the best defined, microRNA-independent role of Drosha that is known. If we then look more specifically at what we've pulled out in terms of the non-microRNA-related mRNAs, we can see 9, and a mercifully manageable 9 transcripts that are differentially expressed when we delete Drosha and LSKs compared to when we delete dicer. The observant amongst you will have noticed that this does include the aforementioned DGCR8, suggesting that this system, this system is both sensitive and robust enough to pick up direct cleavage targets of Drosha, namely DGCR8, which has been previously identified. In addition, we've also got within this 2 cytoskeletal components, a long non-coding RNA, an antigen-like protein, 2 putative proteins with no known function, a uric transporter, and a phosphatase. So although we've shown that there's increased levels of these 9 transcripts in LSKs, the obvious next question is this increase in level actually relevant and does it impact dendritic cell development? So in order to answer this question, I'd like to just briefly take you through our working model. So what we propose is that in LSKs, Drosha cleaves the mRNA of an anti-differentiation factor. That keeps this factor at low levels, allowing proper differentiation of dendritic cells and myeloid cells. However, upon Drosha deletion, we propose that the mRNA of this factor remains unfettered, uncleaved, and is able to produce its product, which potentially accumulates and blocks proper dendritic cell and myeloid development, leading to the reduced dendritic cells that we observe upon this impact. So in order to determine whether the increased levels of these 9 transcripts are potentially underlying this Drosha deletion phenotype, we used an overexpression experiment. As such, we engineered retroviruses that induced overexpression of the open reading frames of one of each of 9 of these putative Drosha targets. We then hypothesized that if we overexpress these transcripts in a wild-type situation, that potentially this induced accumulation would potentially recapitulate the phenotype that we observe in myeloid cells upon Drosha deletion. So hoping to see a recapitulation of the Drosha phenotype upon overexpression of these transcripts. In order to quickly test this, we simply sorted wild-type LSKs and then infected those LSKs with retrovirus that, as I say, induced overexpression of one or of each of the open reading frames of these identified putative Drosha targets. We then again put these into FLT3 ligand culture, which, as I suggested, is a growth factor that induces dendritic cell development in vitro. And then after 8 days, did a FACS analysis to determine the percentages of DCs in these cultures compared to control. And in fact, very excitingly, what we observed was that the overexpression of just 2 of these putative Drosha targets did have an impact on in vitro dendritic cell development. That is, that if we examine the percentage of the CDCs in culture that overexpress MIL9 or 26103 118 NO2-RIC, or NO2-RIC for just simplicity and so my tongue doesn't fall off, we can see there's a significant reduction in the percentage of these DCs when we overexpress these guys, suggesting potentially that the overexpression of MIL9 and NO2-RIC upon the deletion of Drosha is potentially underlying the phenotype that we observe. Importantly, in addition, we saw no impact when we overexpress the 7 other putative Drosha targets in this system, suggesting potentially that the increased levels that we observe by microarray in LSKs of these guys is potentially unrelated to dendritic cell development. While this reduction in DCs in these cultures is not as dramatic as the reduction we observe when we delete Drosha, this is to be expected, as we well know that the deletion of Drosha leads to a number of changes, and the causative mechanism, there's an accumulation of causative mechanisms, one of which is the loss of microRNAs. However, we propose that part of this reduction is caused by potentially the de-repression and potential accumulation of MIL9 and NO2-RIC. So in order to determine whether this phenomenon we observe in vitro was also true in vivo, we again sorted wild-type LSKs, and in this instance infected them with retrovirus that induced overexpression of just Mil9 and NO2-RIC. We then put them into a lethally irradiated recipient, again which has the hematopoietic system completely ablated by this irradiation. Left them for a short of 14 days and then examined these cells in the spleen at that time point. So if we simply collagenase digest these spleens again and look using Siglec-H and CD11C by FACS analysis, we can see that approximately 3% of these spleens are PDCs and approximately 6% are CDCs. However, if we examine the fraction of the CDCs that are overexpressing Mil9 and NO2-RIC in vivo, we can see a dramatic reduction in both the percentages of PDCs and CDCs in DCs that overexpress this Mil9 and NO2-RIC, again suggesting that we get a recapitulation of the phenotype we observe upon Drosha deletion. This is quantified here, and again you can see a significant reduction in both percentages of CDCs and PDCs in those that overexpress Mil9 and NO2-RIC. This is not, again, similar to the effect we observe upon Drosha deletion. This is not restricted to dendritic cells in that we see a reduction also in the percentage of myeloid cells and monocytes that overexpress Mil9 and NO2-RIC. Thus, what we can say is that we do see a substantial recapitulation of the phenotype that we observe upon Drosha deletion in myeloid cells in a similar manner to when we overexpress Mil9 or NO2-RIC in these cells. So the obvious question, besides a very short introduction, is what are Mil9 and NO2-RIC? So Mil9 is a cytoskeletal component, as I mentioned earlier, but has not been shown very much to have a role in myelopoiesis. In fact, it's only really been shown, it has been shown to be involved in cell locomotion, migration, and in fact cell signaling. In terms of what's known in myelopoiesis, it's been shown to be involved in the formation of platelets and to be upregulated in vitro in terms of macrophage and monocyte differentiation in humans. That's all that's known. While the limited knowledge that we know about Mil9 in myelopoiesis, that's a veritable library, given what we know about NO2-RIC in myelopoiesis. In fact, we have no known role of NO2-RIC in myelopoiesis, and in fact in any other process. It's a putative protein with no known function, and in fact no clear identifiable domains. However, it has been shown to be conserved across mammals and in fact in lower organisms, potentially suggesting some kind of at this point unknown functionality. So up to this point, we've been proposing that Mil9 and NO2-RIC are cleaved by Drosha, thus regulating them in LSKs. However, we haven't in fact shown that this cleavage does occur. So the obvious question is, are Mil9 and NO2-RIC cleaved? In order to answer this question, we used a relatively elaborate molecular technique, some components of which you guys may know through as degradome analysis or race sequencing. Effectively, what we did was to take total RNA from dendritic cell progenitors. We enriched these for a poly A, and then we specifically and exclusively ligated an adapter of known sequence to those transcripts that were exhibiting a monophosphate on the 5' carbon of the terminal nucleotide. And that 5' monophosphate is a molecular signature of endonuclease cleavage, such as that by Drosha. This 5' monophosphate is not produced by, for example, RNA degradation simply, or by non-specific nuclease cleavage, such as that by another non-specific nuclease. This allowed us to effectively tag 5' monophosphate or endonucleolytically cleaved transcripts using this adapter of known sequence. We then run a reverse transcription, and then from this complementary DNA, we ran a series of PCRs, all of which were, in the first instance, primed from our adapter of known sequence, which will only be present if our mRNAs of interest have been cleaved, in this instance, Mil9 and NO2-RIC. However, the other primer was one that were designed to target a series of locations within our mRNA of interest. That is, again, Mil9 and NO2-RIC. If cleavage was present in these transcripts, we would get PCR products such as this. That is, primed from our forward adapter, but also from regions within our transcript of interest. We could then Sanger sequence these products And this allowed us, by looking for the junction between the adapter sequence and that of the transcript, that allowed us to identify the original position of the 5' phosphate and therefore the original position of cleavage. Using this technique, we identified a single cleavage site within both MYL9 and NO2-RIC, and we can see those here. And they're all both within the exons of NO2-RIC and MYL9. If we focus more specifically on those regions of cleavage, we can see that in the case of NO2-RIC, a very nice stem loop structure forms if we analyse them for thermostability. That is, just simply looking at the sequence and predicting a sequence that will a secondary structure that will form based purely upon that sequence. So we can see a very nice stem loop structure potentially recognised by Drosha. The MYL9, the same thing. Around the region of cleavage, there is a stem loop, a potential double-stranded RNA stem loop structure. This is just an example of the stem loop structure that's been previously identified to be cleaved by Drosha in DGCR8 mRNA, and as you can see, a very good likeness. And all three of these, again, have a very good likeness for those stem loop structures that are recognised in primary microRNAs by Drosha. So while we've shown there is cleavage in MYL9 and NO2-RIC in dendritic cell progenitors, and we've shown that there's double-stranded RNA stem loop structures that are potentially cleaved by Drosha, we haven't in fact shown that this cleavage is dependent on Drosha, and so that's the obvious next question. In order to do that, we returned to our molecular technique and slightly modified it. In this instance, we took our total RNA from control or Drosha-deleted dendritic cell progenitors. Took it through the regular process, and however, in this instance, instead of running the tiled PCRs, we simply ran a qPCR using the primers previously used to identify the cleavage site. And what we showed was that in Drosha-deleted DC progenitors, there is significantly less cleavage of both MYL9, NO2-RIC, and in fact DGCR8 upon deletion of Drosha, suggesting that in very large part, the cleavage of MYL9 and NO2-RIC is performed by Drosha. So, in conclusions, what I've shown you today is that the deletion of Drosha blocks dendritic cell development and that this impact is independent of microRNAs. Furthermore, we've shown that deletion of Drosha in LSKs causes an accumulation of a number of mRNAs. Two of which, namely MYL9 and NO2-RIC, repress dendritic cell development both in vitro and in vivo. So potentially the upregulation of these guys upon the deletion of Drosha is part of the underlying mechanism observed upon Drosha deletion. Furthermore, we've shown that this cleavage is in fact performed by Drosha. And as such, this has identified the first microRNA-independent function of Drosha with a physiological role during hematopoiesis. And as such, what we propose is that this microRNA-independent role of Drosha is forming some kind of a regulatory mechanism specifically in HSCs, or hematopoietic stem cells. And this would be consistent and fits quite nicely with the broader picture of the microRNA-independent roles of Drosha. That is, the majority of previously identified microRNA-independent roles of Drosha has been identified predominantly in stem cell populations. As such, if we delete Drosha and DICER in mature cell types, such as T or B cells, we see a similar, if not identical, phenotype between the deletion of Drosha compared to the deletion of DICER. However, if we delete Drosha in, for example, neuronal precursors, we see a loss of stem cell fidelity, but there's no phenotype when we knock out DICER, suggesting that these microRNA-independent roles occur predominantly in stem cell-like populations. If we look more specifically at the impact in neuronal precursors or progenitors, it was shown that the underlying mechanism of this impact was similar to what we propose in hematopoiesis, in that Drosha was shown to cleave double-stranded RNA stem loop structures in the messenger RNA of a proneuronal transcription factor called neurogenin 2. Shown here. And as such, what was shown is that under normal conditions, under wild-type conditions, Drosha cleaves the mRNA of this proneuronal transcription factor, keeping it at low levels and thus maintaining the stem cell-ness of these neuronal progenitors. However, upon the deletion of Drosha, neurogenin 2 is de-repressed, accumulates, and plays its proneuronal function in that it leads to precocious differentiation of these neuronal stem cells. The physiological consequence can be seen here, in that under control conditions, when Drosha is controlling the levels of neurogenin 2, we see a very nice structured and organised mouse forebrain in that you can clearly distinguish the ventricular zone and the subventricular zone. However, upon deletion of Drosha, neurogenin 2 accumulates, plays its proneuronal function, and leads to this completely disorganised neuronal forebrain, this mouse forebrain that you can see here. Again, returning to our stem cell-like populations, in contrast to the precocious differentiation observed in neuronal progenitors, if we delete Drosha in embryonic stem cells, for example, it's been shown that these are refractory to differentiation stimuli, in that they no longer differentiate as they should. So what the work I've shown you today and performed as part of my PhD allows us to add hematopoiesis to hematopoietic stem cells to this list of stem cells that are affected by a microRNA-independent role of Drosha. And as I said earlier, this has allowed us to identify the first microRNA-independent impact of Drosha during hematopoiesis. So, a PhD is not a journey that can be taken alone, and as such, there are a large number of people who deserve acknowledgement here. First of which are Andrew, who you've met, Ifan, and Mark. They're my three supervisors, and I thank them very much for their excellent and dedicated supervision over the last 3.5 years. In particular, I'd like to thank Andrew for his exceptional knowledge of the past, present, and in fact future of the field that we call immunology, and the world more broadly. I thank Ifan for sharing his serenity and his very practical shortcuts, and Mark for, again, his exceptional practical experience and sharing of that, and his exceptional attention to detail. More specifically, I thank all members of both labs, past and present, who've made both labs an enjoyable place to work. I also thank the Immunology and Molecular Immunology divisions more broadly, again, for making Level 3 a very enjoyable and exciting place to work. In particular, I thank the Shortman Lab, that is David Remick and Lauren Cox, for the very generous sharing of excellent reagents with which a large part of this dendritic cell work would not be possible. In addition, I thank Yuja and Gaetano for work I haven't presented today, but they've also been exceptionally helpful. Bioservices, or whatever they call themselves now, we are extraordinarily lucky here at WEHI to have access to the number of facilities that we do. Every time I didn't have to irradiate my mice, every time I didn't have to make my own DME, every time I took a beaker off a shelf and then chucked it away dirty and I didn't have to clean it, I was thankful and continue to be. So that they definitely deserve acknowledgement. I'm thoroughly underappreciated. I also thank Catherine Yates, or Nee Yates, I should say, hey more specifically, and Merle for their excellent mouse assistance. Without their expertise and dedication, long, hard days would be significantly longer and harder, so I thank them. The FACS facility, all of our immunology projects require a large amount of sorting. They've been exceptional, and when I began, I was sorting my own cells, and it is not a process I wish to do throughout my PhD, so I thank them. Friends and family, a large number of these people have endured far more of this process with me than they deserve, and I thank them for it. The NHMRC for my scholarship up to very recently. The University of Melbourne for mostly letting me just get on with it. The Walter and Eliza Hall in two parts. First of all, for the Edith Moffat Scholarship that's allowed me to complete my thesis in a timely manner, but also because I found this place an engaging, dynamic, and exciting place to work. And for that, I thank you, the engaged members of the WEHI community. Shame on all those who are absent. And then finally, and as always, I thank the mice that paid the ultimate sacrifice to bring light to these particular truths. I thank you again, and welcome any questions that you might have. Farah, you want to do it, Andrew? You want to drive? Farah.
I've got 2 questions.
Lovely.
First one, general, but you said that Drosha sometimes recognizes messenger RNA these stem loops, and
I hope it does, yeah.
and sometimes it stabilizes them, or sometimes it causes them to be degraded. So what is it? Is it something about the stem loop structure that determines the outcome?
Right, so I may have misspoke, or you may have misunderstood. Always cleavage.
Okay.
Yeah, and the structure is very important. So, in fact, the stem loop structure is recognised by the aforementioned DGCR8. So Drosha doesn't necessarily recognise it alone, as with most proteins, it acts in a complex. And DGCR8 positions it very specifically on that stem, but always cleavage. Yeah. Second part?
The same question was, you said that Drosha probably plays its non-microRNA roles in stem cells. I don't know if you mentioned this, but does that mean that its levels decrease as cells differentiate?
An excellent question. I suppose the basis of that is why would Drosha play this microRNA-dependent role specifically in stem cells? The short answer is we don't know. The long answer is that it's probably cofactors that, for example, are interacting with Drosha in a complex that we don't yet know about, or cofactors that are acting on the mRNA specifically in hematopoietic stem cells that lead to the cleavage specifically in stem cells. So, for example, MYL9 is a cytoskeletal component, so it should be expressed throughout. Why it's cleaved specifically in hematopoietic stem cells, we don't know, but cofactors is the short. It's the long.
Angus?
Gus.
Congrats, Tim.
Thank you.
I just got a question about your dice knockouts. Did you ever try overexpressing MYL9 or an O2 RIC in those and seeing if you got the same phenotype?
Great question. No, great question. On the to-do list, near the top, but a great question.
This is maybe a naive question, but I guess DICER and Drosha are obviously very important. When you're looking at just cell surface markers that tell you what a specific type, that's how you define what a cell is. How much do you affect that? If you just put a protein that's supposed to just go to the surface and delete Drosha and DICER, do you see an increase or decrease? Do you know what I mean?
I know the first part of what you mean. In that, how does deleting Drosha affect? We're only looking at the surface. We're looking completely at the surface. Yeah. I agree that it's a downfall. However, in this instance, what we're really trying to pull out is PDCs and CDCs.
Yeah.
We don't see any change in the levels of Siglec-H or CD11C, or in fact a number of other markers that we've used to determine that those are PDCs and CDCs upon the deletion of Drosha or in fact DICER. So in this instance, it's not affecting the levels of Siglec-H or CD11C or any of the other guys that we're using to pull them out. I can't say anything about other surface markers it may well be affecting.
Yeah.
But
also with Drosha, you showed that MYL9 and RIC get cleaved,
yep.
but only half of it.
Yeah.
Do you have any idea what's...
Good point, an excellent point. I suspect that the short answer is I suspect that it's potentially Drosha protein residual after the 72 hours. So although we looked at the deletion of transcript of Drosha, it's potentially possible that protein is sticking around. So that may be why some of it's not lost. Background is the other short answer. Yeah, so I suspect residual protein, I suspect.
So your rationale for looking for microRNA independent function was that the Drosha phenotype was more severe than the DICER phenotype. Am I right in thinking that we're talking about DICER1?
Yes. Yeah.
And what if you incorporate DICER2 in the whole argument?
Yeah.
You would have to look at DICER1 to double knockouts.
I don't know. We haven't explored that.
Because you could have a situation where double knockout of DICER1 and DICER2 actually moves exactly by proportion.
Potentially, the microRNA-independent role of Drosha that we've identified would still be a function. Would you not agree?
Potentially, I'm not saying it's not
Yeah.
potentially, because you still, by showing what you've shown, you still could be secondary to other effects. Because you have to actually...
some parts might be secondary. I don't think that the cleavage of these stem loop structures by Drosha, there's no way that can be secondary. In that we've shown cleavage of these two structures by Drosha. I agree that the way in which we have got to that point, by for example, not knocking out DICER2.
When your argument that it's Drosha is really Drosha negative and positive cells.
Yes.
That's really simple. That's not, I'm not saying, I'm not trying to say that that's not potentially direct, but it could also be indirect. You could have an effect on, you could still have an effect on microRNA cleavage of this particular product, mRNA by another.
Yeah, I suppose you're right. The way in which we're going to answer potentially part of that question is to show in vitro that Drosha does in fact cleave these stem-loop structures. So we're going to, the idea is to isolate Drosha by precipitation and then mix that in vitro with these stem-loop structures that we've cloned. It's starting tomorrow, in fact. So that's the only way that we can answer that question. Keep drawing, Andrew.
How is that?
How conserved are the cleavage sites amongst species in the two target RNAs that you have found, is the first question. And the second is it conceivable that the change in cleavage efficiency of the mRNAs affects other mRNAs? So when you overexpress the two mRNAs, have you done microRNA RNA analysis expression in the cells that overexpress these two RNAs?
The answer to the two questions is no. I haven't looked at the conservation of the specific site within other species, which is a great idea. MYL9 is well conserved across species. That's what I have looked at. I haven't looked specifically at the site, so I would assume that it's very good. The other question is something I've thought about quite a lot, in that if we overexpress a Drosha target, potentially we're clogging up Drosha's functionality on other things. We haven't looked at the impact of overexpression of MYL9 and NO2-RIC on microRNAs, for example. That's something, again, on the to-do list. The only answer that I've sort of thought of is that if that was the case, that the overexpression of MYL9 and NO2-RIC was disrupting microRNAs, then we would expect to see the phenotype when we overexpress some of the 7 other targets, in that the overexpression would be similar, potentially clog up Drosha, and then we would expect to see a phenotype because of that clogging.
Yeah, I was not thinking of an artifact due to overexpression.
I'm sorry.
But whether in fact a target of Drosha could specifically, could play a physiological role in controlling the RNAs, so that the phenotype that you see is not due to the protein, to changes in expression of the protein that is targeted to, but to changes in expression of other proteins that are affected by the changes.
Yes, yeah. We haven't looked. The only way that I've thought of dealing with that is to further examine the MYL9 and NO2-RIC overexpressing cells. So, for example, do further analysis on those to determine the effects of simply the overexpression of those kinds.
So you must have also spent quite a bit of time thinking about what to do with such unpromising names as MYL9 and
Wouldn't go so far, but yes.
Could possibly be doing to impede dendritic cell differentiation.
Yeah.
So, first of all, your ideas, but does the primary sequence of the RIC cDNA give you any clues on the name structure?
No.
Anything interesting there?
In terms of NO2-RIC, the short answer is no. There's nothing identifiable in there that would suggest some kind of role. So the only way we're going to get at that is further examining, as I suggested, the overexpression. MYL9, the mind wanders. The first thing I thought of was, because it's a cytoskeletal component, because it's involved in migration, potentially it's impacting something around the niche, stem cell niche. Why that would, how that's playing a role, I don't really know. Perhaps it's holding these stem cells in the niche for longer than they should be in there, and therefore they're getting additional signals. The short answer, as I said, is we don't really know. Those are the things that I've been thinking about. The only way to get at them is to further examine the HSCs that are overexpressing the 9 and We're going over, and careful now. Anyway, sorry. Go on, Andrew. Is it? Andrew?
Yeah, that's right, yeah. So there's lots of different kinds of small RNAs now, sort of defined by their biogenesis. So I'm sort of wondering if you know what happens to the hairpins once they're clipped? The NO2 hairpin and the MYL9 hairpin?
Yeah, it's a great question. Mostly they're degraded. However, you can pick them up by deep gene sequencing. So you can pull out these guys. Sometimes they go on and to be processed by Drosha. by DICER. So we can pick these guys up. but that's only a recent thing well, relatively recent with the onset of deep gene sequencing. In fact, we used that characteristic, that ability to pick them up, to confirm these cleavage sites. So we found small RNA fragments that are traces of Drosha cleavage derived from these sites. So you can pick them up, but they're obviously going to be quickly degraded, and that's the purpose of it. But you can pick them up. Yeah. Given it's 4:02, Andrew, I'm going to take control. Yeah. Thanks, everyone. Thank you.